Perkins. Brian, perhaps remember many years ago, we had a weekly in-person meeting. It was local, only for people at the University Hospitals, Cleveland Clinic, and CWRU. And this meeting now grew to quite international show. So we're very delighted to have you and greatly appreciate dressing up for us. Although Brian always dressed up properly. With that, let's Yasmin introduce our speaker.
Good morning, everyone. My name is Yasmin Solano. I'm a third-year graduate student within Dr. Philip Kaiser's laboratory. I'm happy to introduce today's speaker, Dr. Brian Perkins. Dr. Perkins received his Ph.D. in biochemistry at Baylor College of Medicine in Houston, Texas. After completing his doctoral studies focusing on the identification of triplex targets in the human rhodopsin gene and how they affect their structure and expression, Dr. Perkins moved to Harvard University in Cambridge, Massachusetts, to pursue postdoctoral training in the field of retinal cell biology with Professor John Dowling, a pioneer in using zebrafish as a model to study development and genetics of the vertebrate retina. Since starting his independent lab in 2004, Dr. Perkins has trained several PhD students, one of those being our lovely Emma Lissier, here at UCI CTVR. He's trained postdoctoral fellows and more than 30 undergraduates. Dr. Perkins' research has pushed science forward in revealing new genetic pathways regulating primary cilium formation and maintenance in zebrafish. His findings have been extrapolated to the retina since the photoreceptor outer segment is considered a modified, highly specialized primary cilium, and so his work has provided novel insights into potential mechanism of blindness. The publication record of Dr. Perkins is extensive and includes several articles and book chapters. He has received continuous funding from the NEI and is one of the recipients of the Research to Prevent Blindness Doris and Joel Steen Professorship. Without further ado, please help me welcome Dr. Brian Perkins.
Thank you very much. Can everybody see my screen?
Please you share again, Dr. Perkins.
Okay. Let's see. Now, can you see it?
Yes.
Okay, good. Now let me get the pointer going. That should work. Move this out of the way. Wonderful. Okay. Uh-oh, let me get that. Zoom is always so much fun. So I'd like to thank Chris and Regeneration for the invitation to speak today. I'd also like to thank everybody for coming and giving me the opportunity to discuss some of the work out of the lab. And so over the next 45 to 50 minutes, I want to present our results from ongoing efforts in stimulating photoreceptor regeneration in a zebrafish model of blindness. Cool. We're going to advance. Uh-oh. Why is it not advancing? There we go. Okay. So I'll start first by acknowledging the people who did a lot of the work. Really the first part of the talk will focus on work that was the PhD dissertation work of Emma, your colleague, with some help from a project scientist, Ting Song, as well as a former project scientist, Joe Fogarty. The remainder of the talk will focus on some published and unpublished work from Ting, Joe, with contributions from Sarah Grabinski, who is a former senior lab technician, and Dwani Prasanna, who's a current tech in the lab. And all of these are fantastic colleagues, and honestly, the work was not going to get done without them. They've been fantastic in the lab. So this is sort of the roadmap of where we're going to go over the next 45 minutes to an hour. I'll give you an introduction and some background, go into some of the fish models of retinal degeneration, and then go into the real meat of the talk, talking about regeneration. So I started my lab about 20 years ago with an interest in the cell biology and genetics of inherited retinal dystrophies. And as all of you likely know, what typically happens in these diseases is that the rod photoreceptors in the periphery of the retina degenerate, and this degeneration progresses towards the center until many people only have a small island of cones remaining in a region called the fovea. And eventually even the cone photoreceptors die and people lose all functional vision. And the speed that people lose vision can vary. And so some diseases like retinitis pigmentosa may not start until adulthood, whereas other diseases like Leber congenital amaurosis and choroideremia can start very early in childhood. And these affect thousands of people worldwide. There are more than 300 genes associated with inherited retinal dystrophies with thousands of individual mutations. Now, despite this, all of our knowledge over the last 20 to 30 years that we've gained, the treatment options are still extremely limited. And as I'm sure you all know, Chris's group is doing pioneering work to exploit gene editing technologies and prime editing to correct mutated genes, but there's still obviously a lot of hurdles. And any personalized medicine approach would still require developing error-free approaches that can target and correct unique mutations that cause these disorders, and then work through all the bureaucratic red tape of the FDA. So as an alternative to this approach, there has been certainly an emphasis in regeneration of lost retinal neurons using endogenous sources. And about a decade ago, the NEI introduced this program called the Audacious Goals Initiative. And one of the goals was to push the boundaries of regeneration within the eye and the visual system. And particular emphasis was placed on regenerating photoreceptors for the treatment of inherited retinal diseases. And so the goal of regenerating photoreceptors in humans sounds a bit crazy, but it emerged from knowledge that some species already had the ability to repair damaged photoreceptors. And building off of initial studies in goldfish in the 1970s and 80s, a small number of groups began investigating tissue regeneration in zebrafish in the early 2000s. Now, zebrafish are small freshwater fish. They reach about 1.5 inches or so to 2 inches maybe in size. They're relatively inexpensive to maintain. This is the Lerner Research Institute, of which I'm a part of, and our fish facility is located, way back here in a corner. And Emma probably recognizes this image. This is part of our zebrafish facility, hundreds of fish tanks with thousands of fish. And the current capacity of our zebrafish facility is for about 800 tanks with probably about 16,000 to 20,000 adult zebrafish. And in fish, there's about 70% of the human genes are found in the fish genome. They have similar anatomy and physiology to humans. But importantly, fish have the ability to regenerate damaged tissue in the retina, brain, heart, pancreas, and appendages such as fins. There we go. So, as I said, teleos fishes, including zebrafish and goldfish, have this ability to regenerate damaged neurons. This was first discovered back in the late 60s when they surgically removed part of the retina from a goldfish and saw that it regenerated. Since then, groups have used cytotoxic lesions, light damage, laser damage, and even mechanical injury, needle pokes, things like that, to trigger this regeneration. And so following injury, Mueller cells will undergo a reprogramming event that triggers an asymmetric cell division. So seen here, this Mueller cell will become activated, undergo an asymmetric cell division to produce a new Mueller cell and a neural progenitor. And this progenitor continues to divide and will ultimately differentiate into all types of retinal neurons. It's also important to note that there's a second population of precursors that exists within the retina, in a fish retina. And this population is called the rod precursors. So the rod precursors are also derived from Mueller cells, but these are unipotent and only give rise to new rods. Whereas the Mueller glia-derived progenitors can produce all the other types of neurons, including cones. So what drives this process? So acute injury triggers both morphological and transcriptomic changes in microglia. And in particular, microglia will express and secrete several pro-inflammatory cytokines. And work from several groups has shown that if you immunosuppress the retina by pharmacologically depleting microglia or by treating the animals with dexamethasone, you can inhibit the Mueller glia from proliferating after an acute injury. However, work from a few years ago from Nicholas Silva and Peter Hitchcock found that prolonged inflammation will cause an excess of proliferating progenitor cells that ultimately fail to differentiate properly and they die. And so this also leads to a failed regenerative process. In other words, the retina needs to find this Goldilocks level of the right balance of inflammation between having just enough, whereas too little blocks proliferation to start, and too much inflammation leads to subsequent death. And so we have to find this right amount. Now, our knowledge of regeneration has generally been limited to studies of acute injury in wild-type fish, and how regeneration is regulated in models of degeneration has largely been unexplored for the time being. So this is kind of an obvious gap in knowledge that we tried to step into probably about 5 or 6 years ago now. And we were interested in how regeneration might occur in a model of progressive retinal degeneration. And so to address this question, we spent the past several years now developing and characterizing a number of fish mutants with slow progressive photoreceptor degeneration. And we first started with a fish line with a mutation in the gene encoding CEP290. And as I'm sure many of you are aware, mutations in CEP290 lead to retinal disease in humans. CEP290 is a component of the ciliary transition zone, which is located right here. Really, the transition zone is where the connecting cilium is, although the axoneme extends up through the outer segment. And this transition zone serves as a diffusion barrier to prevent the wrong sorts of proteins from entering into the outer segment. Work in worms and a little bit in mouse has found that CEP290 also coordinates transition zone assembly during development. And the mutations, obviously, in CEP290 are important because they cause multiple forms of retinal disease. Now, the knockout mouse models of CEP290 are frequently embryonic lethal, and they don't always basically recapitulate disease because obviously humans frequently will form functional photoreceptors that then subsequently degenerate, whereas in CEP290 mutant or knockout mice, they frequently fail to form photoreceptors at all. Now, the fish mutant was identified about 15 years ago in a genetic screen for mutations affecting early development. And the identified mutation introduces a stop codon roughly halfway through the reading frame. And this mutation is predicted to truncate the protein upstream of this microtubule binding domain. And Emma found during her early characterization that it reduced mRNA expression by about 50%, likely through nonsense-mediated decay. We were never able to obtain a really good antibody, so it's possible that there is still some protein being produced, but we don't really have a way of testing that. The fish mutants are viable into adulthood, although they do develop the scoliosis phenotype that you can see just in the adult fish as well as in micro CT scans. And this phenotype is observed in many fish cilia mutants that are able to reach adulthood. So when we look at the retinas for evidence of retinal degeneration, we stain the retinas with a couple of markers that are widely utilized in fish research, as well as retina research in general. So one is peanut agglutinin, or PNA. This stains the extracellular matrix around the cone outer segments. We also stain with a marker called ZPR1, that is a marker for the cone inner segment. And so this top row shows wild-type animals at 3 months or 6 months of age. And you can probably appreciate that even at 3 months, the PNA and ZPR1 staining in our CEP290 mutants is already a little bit disorganized. By 6 months, the number of cones is already significantly declined. And by 12 months, you can see that there are very few cone photoreceptors left after about a year. And so we've quantified that over time, and you can see that even at 3 months, you see about a 20% reduction in the number of cones, and by a year, you've lost probably 80%. And here at the 6 month time point, which is what we use for most of our studies, the animals have lost somewhere between 40 and 50% of their cones. So what about the rods? Well, the rods also exhibited signs of photoreceptor dystrophy, but this appeared at slightly later ages. So this shows retinal section stained with an antibody against rhodopsin. And this labels obviously the outer segments. At 3 months, we don't see any loss of rhodopsin staining, and we also do not see any evidence of mislocalization into the inner segments or the outer nuclear layer in the mutants. However, at 6 months, you can see already that there's considerable mislocalization of rhodopsin. Obviously, the majority of the protein is still located in the outer segments, but you're seeing signs of mislocalization, which is indicative of unhealthy rod photoreceptors. By 12 months, you still have very robust outer segments. There's still mislocalization, but importantly, you can also see that the gap between the outer nuclear layer and the base of the rod outer segments has decreased here, noted by this bracket. This is where the cone outer segments typically reside. This is kind of secondary evidence that the cones are degenerating and that space is being decreased. Importantly, however, and interesting, was that we did not observe a net loss of rods. In spite of this mislocalization, there wasn't a lot of evidence that the rods were actually degenerating. And so I'm going to come back to that in a few slides. As inflammation is believed to drive this regenerative response in zebrafish, we then asked if microglia and other macrophages were activated in the mutants. And so we look for a large inflammatory response noted by the presence of activated microglia. And in wild type, you can see this is a low magnification view stained for 2 markers, 4C4, which is recognized as an epitope on a galectin-3 binding protein, and L-plasmin, which is a pan marker for macrophages and leukocytes. And so you can see that these overlap perfectly within the retina. And these microglia are located within the plexiform layers of the wild type animal, with some also present in the subretinal space. You can see here on slightly higher magnification here. In the mutants, however, you see a large accumulation of activated microglia out here in the subretinal space. And we've quantified that difference here, and we see an increase in the number of 4C4 positive cells. There's also, interestingly, a decrease in the number of microglia within the inner plexiform layer, with no change in the outer plexiform layer. Now, we don't really know if this reflects a migration of microglia between layers. It's not really clear. We don't have the ability to do any sort of live imaging with this. Importantly, though, in spite of the inflammation, the mutants did not exhibit considerable Mueller cell activation. And so we wondered if the mutants had lost the ability to respond to damage. Because as I showed you, these fish undergo a slow degeneration with elevated inflammation. But I also spent the first part of the talk, trying to convince you that fish regenerate, due to inflammation. So we started to look a little bit more about that and wondered whether the mutants exhibited signs of inflammation, what might be needed to enhance this regenerative process. So it's important to consider, whoops, go back one. It's important to consider how we assess regeneration. Now, a fundamental aspect of the regenerative process in zebrafish is cell proliferation. So to monitor cell proliferation, we utilize PCNA immunostaining and the use of the thymidine analog EDU, and each has its own certain advantages. So PCNA is a cofactor for DNA polymerase. It's expressed throughout the cell cycle, starting as early as G1 and staying expressed through mitosis. Now, so when a cell enters the cell cycle, PCNA is going to be expressed, and this can be measured by immunohistochemistry. Importantly, this only will detect cells that are in the cell cycle at the time that the eyes are harvested and the fish is sacrificed. If it's later than that, the cells might become post-mitotic, go into G0, and PCNA will no longer be expressed. EDU is a thymidine analog that's going to be incorporated into newly synthesized DNA. And if EDU is present when a cell is synthesizing DNA during S phase, the EDU will become incorporated into the new DNA, and this will mark the cell permanently. Even if the cell becomes post-mitotic, that EDU can still be detected days, weeks, or even months later because it's embedded within the newly synthesized DNA. And this allows us to then track cells that have proliferated during a time when EDU is present, even several days or weeks later. So to start, we first looked for evidence of regeneration by staining retinas for PCNA. At 3 months, you see a few PCNA positive cells in the outer nuclear layer. You also see a few in the internuclear layer. Same result was found in CEP290 mutants at 3 months. By 6 months, however, the number of PCNA positive cells has largely decreased to very, very few, only just a handful per section. There is a subtle increase in the number of PCNA positive cells in the internuclear layer in our mutants. We think that these are Mueller cells that are proliferating to generate new rod precursors. The reason we think that is because the number of PCNA positive cells in the outer nuclear layer is up by nearly tenfold. And notice the scale on the y-axis here. We have probably tenfold more PCNA positive cells in the ONL than we do in the internuclear layer at this time point. We think that the Mueller cells are generating new rod precursors. These are then dividing to make new rods, which is why we don't see the net loss of rods in spite of the rhodopsin localization and likely rod photoreceptor death. So this made us wonder whether the Mueller glia have simply lost the ability to regenerate at all. The rod precursors clearly can respond to degeneration, but the Mueller cells are not. In order to address this question, we simply challenge the animals with a light damage paradigm. So with this, we put fish in a beaker and we shine light from a fiber optic connected to our microscope, basically illuminate them with really high intensity light for about 50 minutes to an hour. And we then subsequently expose them to high intensity white light for another 5 hours. At roughly 48 hours after injury, we'll inject the animals with EDU. A subset of the animals will be sacrificed at 3 days post-injury to simply confirm that damage actually occurred. The remaining animals are allowed to recover for up to a month, where we'll then assess regeneration. So in response to acute damage, we can see a significant proliferation of the Mueller cells. So here, following our light damage paradigm, we did this in wild type or in our CEP290 mutants on a transgenic background that's expressing GFP within the Mueller cell, so we can co-label the Mueller cell marker with, in this case, EDU. And so you can see that there's a considerable increase in the number of EDU positive cells in the INL. These co-localize perfectly with the GFP signal, indicating that these are proliferating Mueller cells. We also see this in our mutants down here. We've quantified this, and we see equivalent numbers of EDU positive cells in both the internuclear layer and the outer nuclear layer at 3 days post-injury. This told us that the Mueller cells in our CEP290 mutants are equally capable of responding to acute damage and proliferating. It did not tell us anything about whether these could regenerate into actual photoreceptors. So we let the remaining fish grow up. And so what this shows, this panel or this slide will show you that regeneration can occur. So the top row here, the left side are cones, the right side are rods. And what the top panel shows are uninjured animals. These are all 6-month-old animals. So you can see again, PNA in yellow, ZPR1 in cyan. You can see that there's fewer cones already in our CEP290 mutants. You can see the EDU labeling right here. This is staining for rhodopsin in wild type and the mutants. 3 days after injury, you can see that the cones have been completely ablated. The rods have also been completely ablated. But 30 days later, you can see that there is regeneration of both cones and rods noted by the PNA, ZPR1, or the rhodopsin staining. So these data indicate that CEP290 mutants retain the capacity to regenerate. They're just simply not doing it without being injured. So we wondered, however, if given the injury model, whether this regenerative response was capable of restoring the normal density of cones. So this gets us into some kind of esoteric questions a little bit, but also very important ones about what are the limits to regeneration. So consider this example. In an older CEP290 mutant, you may only have about 50% of the photoreceptors remaining. And if you do a light damage, you're going to ablate a large number of those photoreceptors. And so then what happens? We know in a wild type animal, if you ablate photoreceptors, they can regenerate and fully restore the cones back to the same density and the same number of cones that were present prior to injury. So in CEP290 mutants, do they regenerate the full complement, or do they regenerate that's something less than, maybe even equivalent to where they were when they started? So again, we do the same sort of injury paradigm where we illuminate the fish, with a high intensity light followed by additional white light. We label the animals with EDU, allow them to regenerate for up to a month. This EDU pulse will allow us to track where the light damage occurred, because the proliferating Mueller glia will localize to the site of the light damage. So one of the things that occurs during these light damage experiments is that the injury only occurs within about the central third to half of the dorsal retina. And this has been well established by several groups. Now, this is a nice advantage because we can identify the lesion area by staining for EDU and then observing for the presence of proliferating cells that have been migrated and then become incorporated into the outer nuclear layer. And so these dotted lines mark the site where the injury occurred. Interestingly, however, this also provides you with a nice internal control of what the photoreceptor density was in the same animal, because this region out here in the periphery was not damaged by the light. And so these cones are the original cones, whereas these photoreceptors here are those that have been regenerated following light damage. So what we can do is quantify the cone density within the region that's been regenerated and compare it to the cone density in the uninjured area of either wild type or our CEP290 mutants. And so we've quantified this all down here at multiple different ages. So it's probably easiest to see at 6 months. So if you look at a wild type, the surround represents the uninjured original retina. And so this is the cone density here. This is the cone density of fish of the injured area within the regenerated part of the retina in a wild type animal. This is the surround or the uninjured cone density in a CEP290 mutant. You can already tell that it's lower than the wild type, which is normal because it's been degenerated. The lesion area, however, is the regenerated retina, and there's no statistical difference between the cone density in the regenerated area versus the uninjured area. And both of these are significantly different than the wild type. And this result held true across all the different ages. So while cones could regenerate, they only regenerated back to the density that was present at the time of the lesion. They did not regenerate up to wild type levels. And this appears to hold true regardless of age. And importantly, the ability of the wild type animals to regenerate did not appear to decline with age either. So even really old wild type animals show robust capacity for regeneration. Now, we knew from our immunostaining studies that CEP290 mutants had elevated numbers of activated microglia and inflammation. And over the last few years, the role of inflammation and microglia within the regenerative response has come under really intense investigation. And so we want to get a little bit better handle on this within our mutants. And so we ran an RNA-seq, just a bulk RNA-seq experiment on retinas from CEP290 and wild type siblings at 6 months of age. And not too surprisingly, among the top upregulated pathways, 7 were associated with increased inflammation. And so we wondered what would happen if we reduced inflammation. So the first thing we did was take kind of a sledgehammer approach and use dexamethasone. So dexamethasone is a potent anti-inflammatory steroid. And so if we pre-treated the animals with dexamethasone, what we found was that, one, we significantly reduced the number of microglia present in the retina. So this is wild type, this is our mutant. You can see that in the presence of dexamethasone, the number of microglia was significantly reduced. Importantly, we also found that we almost completely blocked proliferation. This is primarily in the outer nuclear layer where the rock precursors existed, because we didn't really see many Mueller glia proliferating. And these are all in uninjured animals, I should point out. So from these results, we really verified what was already largely reported, which is that inflammation promotes proliferation and the regeneration of rod photoreceptors. And so blocking this by immunosuppression would suppress proliferation. Now, I told you earlier that some inflammatory signaling was necessary to induce the Mueller cells to proliferate. However, excess inflammation led to an overproduction of the neural progenitors and the subsequent death of these photoreceptors. So it made us question, is excessive inflammation that's present due to chronic degeneration in our mutants actually preventing the full regenerative response in our mutants? So going back to this slide here, if you think about the possibility that CEP290, because it has chronic inflammation, as the photoreceptors die, if you do a light damage experiment, maybe this inflammatory signaling fails to get adequately resolved, and this inflammation is suppressing the full regenerative ability of these mutants. So how would we test this? So what we did was to do our normal light damage paradigm. We would inject with EDU at 2 days after the injury. And starting at 3 days after injury, we would immerse the animals in a dexamethasone solution from anywhere from 9 to 12 days. And then after a couple of weeks, we would then harvest the eyes and look for and measure and assess cone density. So this is very preliminary work. We need to replicate this a couple of times. So if you treat wild type animals and you damage them, I've already shown you this data, which is that the animals, this is in the damaged area, this is in the uninjured area, the photoreceptors regenerate back to normal. In our mutants, in the undamaged fish, you see a decrease in the number of photoreceptors. Without light damage, if you add dexamethasone, you don't really see an effect. However, if you damage the retina and you treat them with dexamethasone, we see an increase in the cone density. Now, I need to point out that these numbers are not statistically significant at this point, but there is an obvious trend towards rescue. Like I said, we need to repeat this a couple of more times. We've done this with some other mutants and seen more significant results. So I feel pretty confident that if we can facilitate the resolution of the inflammatory response, then we should be able to rescue and regenerate more cones in our model. Now, dexamethasone is a temporary method of immunosuppression, and retinal degeneration, particularly in these models, is a chronic condition that's occurring over the course of months. So we wondered if there was a way to lower inflammation for a much longer period of time. And so to do that, we turned to the zebrafish IRF8 mutant. IRF8 is a transcription factor. It's critical for macrophage and microglia development. And we known from previous reports that in adult IRF8 mutants, there is a significant reduction in the number of macrophages and microglia within the CNS. So these are flat-mounted retinas of wild type and IRF8 mutants. Hopefully you can notice the difference here. These are magnification of these areas here. These are all stained with 4C4 to label microglia. We've quantified the number of microglia within these retinas, and we see a significant reduction. Not a complete absence, but it's severely reduced, probably about 80% in these flat-mounted images. So we crossed our CEP290 mutant onto an IRF8 background to make double mutants. So these are staining for microglia, again, our 4C4 and L-plastin staining. This is the wild type sibling. These are our CEP290 mutants. At 6 months of age, you can see all the activated microglia present in the subretinal space. IRF8 mutants, and I should note that all of these are siblings of each other from the cross. IRF8 mutants have very, very few microglia, as do CEP290 IRF8 double mutants. We quantified both proliferation and the number of cones by PNA staining. Suppression or reducing microglia, which would likely cause a reduction in inflammation, also cause a reduction in the number of TCNA positive cells. Interestingly, though, we know that CEP290 mutants have fewer cones. IRF8 mutation does not impact cones at this age. However, in our CEP290 IRF8 double mutants, we see a rescue of the cones. So we have a cone density that is no different statistically than a wild type. So we've actually rescued the cone degeneration by eliminating microglia from the retina. We don't think that this is due to regeneration because we see no evidence of proliferation in the outer nuclear layer or the internuclear layer. We think that this is simply preventing the cones from dying in these CEP290 mutants. So this suggests that the loss of microglia is actually providing a neuroprotective effect against cone degeneration. So we then begin to wonder, what are the long-term effects of microglia depletion on the retina? So looking at IRF8 mutants at multiple ages. So as I mentioned earlier, in a healthy wild-type retina, the microglia normally reside within the parenchyma of the retina, within the outer flexiform layer, the proximal and distal surfaces of the IPL, and within the subretinal space. In our IRF8 mutants, we analyze these at 3, 6, and 10 months of age. We see a significant reduction in all layers of the IRF8 mutant, as well as a total reduction of microglia. But this range anywhere from 80% to maybe as high as 50% reduction, depending on the age and depending on where exactly you were looking. Overall, however, we did not observe a statistically significant difference with age across wild types or across the IRF8 mutants, suggesting the number of microglia remain relatively stable over the course of their lifetime. However, we did notice that in animals that were at least 10 months old, we noticed a small but statistically significant reduction in cone survival. So in our IRF8 mutants here in the open circles, you can see that there's a little bit of reduction in the number of ZPR1 positive cells, a little bit more variation, but a similar decrease in the number of cones when we quantified TNA positive outer segments here. So this just suggests that the absence of microglia or a decrease in the number of microglia will compromise survival of the cones long term. But the obvious, kind of the important question is, what effect does this have on regeneration? So we were very surprised that if we performed light damage experiments on these animals and we quantified TCNA numbers in the internuclear layer and the outer nuclear layer, we saw no difference between IRF8 mutants and their wild-type siblings in terms of proliferation. At 24 hours, 48 hours, or 72 hours, there's no significant difference. There's really no statistical difference at all in the number of proliferating cells between wild type and the IRF8 mutants. Furthermore, if we allowed the animals to recover for a month, we found that the IRF8 mutants fully regenerated the damaged cones. So again, the top panel shows animals prior to light damage. The middle panel shows proliferation and the complete ablation of the photoreceptors here. And roughly a month later, you see full regeneration of cones. And if we quantified cone density after 1 month of recovery, there's no difference in the ability of IRF8 mutants to regenerate. Same numbers as the wild type. So we're still trying to understand and provide a mechanistic explanation for these results. It's quite possible that other cells within the retina will compensate for the loss of microglia and can release pro-inflammatory cytokines in response to acute injury like this. Mueller's cells are known to compensate for microglia during development. Diana Mitchell has shown that. And there may be additional roles present here as well. Alternatively, the few microglia that still exist may be sufficient to induce the Mueller cells to reprogram. I don't believe that our results indicate that microglia are not important. I think they are important for regeneration, but it does indicate that the genetic reduction of microglia does not prevent the retina from regenerating following acute damage. There may be compensatory mechanisms at play. So I want to return briefly to the CEP290 story and hopefully provide a mechanism that can explain why these animals fail to regenerate normally in the absence of damage. So what's preventing them from even attempting to regenerate? So in collaboration with David Hyde and Seth Blackshaw, we conducted a pilot single-cell RNA experiment on undamaged wild-type and CEP290 mutant retinas. And these are done at 6 months of age, again, on undamaged animals. And we obtained reads from all the major cell types, as shown here. And then when we directly queried the Mueller cell population, we found that the expression of Notch3 was elevated in the Mueller glia of the CEP290 mutants. And so we know that downregulation of Notch3 is required for these Mueller cells to proliferate. And that comes from work from David Hyde's group, as well as a few others. And we know that Notch signaling will decrease as inflammation increases. Work from David Hyde found that inhibition of Notch signaling was sufficient to induce Mueller cell proliferation in undamaged retinas. And the idea is that Notch3 will maintain Mueller cell quiescence. So this is work just from in the developing retina. Notch is upregulated early. In the adult retina, if you injure the retina, Notch signaling, especially Notch3, rapidly declines before rebounding back to baseline a few days later. So what we did was we treated wild type and CEP290 mutants with the pharmacological inhibitor of Notch, termed RO4929097, or we treated them with DMSO, which is the vehicle control. So after treatment with the RO drug, even in the absence of damage, we found that there was an increase in the number of PCNA positive cells in wild type animals. In CEP290, we also see a significant increase in the number of PCNA positive cells in both the internuclear layer as well as, particularly in the internuclear layer. Treatment with the RO drug didn't appear to significantly impact the number of PCNA positive cells in the outer nuclear layer, indicating that the ROD precursors don't appear to be affected by Notch signaling. It's predominantly the Mueller cells that are responding to Notch signaling. So what this tells us is that Notch signaling is maintaining the quiescence of these Mueller glia in our CEP290 mutants, and Notch signaling is actually upregulated relative to even wild type. So what have I told you so far today? So in our zebrafish degeneration mutants, these animals are viable. They show progressive cone degeneration. They show signs of rod dystrophy. The rods are likely degenerating, but then they regenerate from the rod precursors. And retinal regeneration in fish requires more than simply neuronal cell death. You have to reduce Notch signaling on top of that. It's not just inflammation. You have to downregulate Notch. And apparently Notch is upregulated above wild type levels in these mutants. So chronic inflammation appears to restrict differentiation and possibly survival of the regenerating photoreceptors after injury. In our mutants, we found that chronic immunosuppression can rescue the cones in our CEP290 model. The reduction of microglia and IRF8 mutants does not appear to have any effect on regeneration. So are there other neuronal sources of inflammatory molecules? Do we possibly need better genetic models to really tease this out to where we can completely eliminate all microglia from the retina? And do microglia ablation paradigms using pharmacology or possibly other approaches, maybe these have unintended or non-specific effects on other cell types? Our results differ from some of the previous reports in the literature, but I think there are probably some good explanations that can explain what these differences are. So what are the next steps? Well, I've told you that the Mueller glia fail to proliferate in our models of degeneration. And it's not just CEP290. We've looked at a number of other models as well, and other groups have reported similar sorts of findings where there is slow progressive degeneration without any evidence of Mueller cell activation. So this seems to be a common theme. Inflammatory signaling remains high. We believe that elevated Notch signaling is preventing the Mueller cells from reprogramming. So does chronic inflammation have a link with this? And does this somehow alter the transcriptome of Mueller cells to resist reprogramming? How does Notch signaling become elevated? What is chronic inflammation doing to the transcriptome of the Mueller cells? How do the regenerated photoreceptors compare to degenerating photoreceptors? A question I get asked frequently is, if you allow CEP290 mutants to regenerate after light damage, do those photoreceptors continue to die at the same rate? Can they regenerate again? We haven't done those experiments, but it's certainly an interesting question. And then finally, how does the inflammatory signaling differ between acute injury versus chronic disease? Are there different types of pro-inflammatory cytokines that are being released in chronic disease versus acute injury? Are they the same? Are the levels the same? Why does acute injury trigger regeneration, whereas chronic disease does not? And our hypothesis moving forward is that in these models, this chronic inflammation is causing some kind of a change, either gliosis or senescence, and this is preventing regeneration. We have a little bit of evidence to suggest that the Mueller cells in these models are just fundamentally different than what you can see in a wild type, or even what you see following acute injury. But we have a lot of work to really follow up on that. And we're using some multi-omics approaches, again, collaborating with David Hyde to look at interrogating these changes in the Mueller cells, as well as looking at microglia and hopefully other cell populations in CEP290 and some other models, both with and without injury. So finally, again, I want to thank members of my lab who really did the bulk of the work. Ping Song is an amazing scientist. He generated most of the data that you saw today. Dewani and Brandy are two technicians in the lab currently who are very helpful. I keep pushing the wrong button. Sarah is a former senior tech that did a lot of work on some RNA-seq that we've done recently. Emily Janik did some work I didn't show you about today, but was critical for understanding the Notch signaling in our CEP290 mutants. Emma was a fantastic graduate student that really set us off on this by working on CEP290 and developing some of the other models I didn't tell you about, as well as Joe Fogarty. We've had a lot of really great assistance and collaborations with people within the vision community: David Hyde, Seth Blackshaw, Jeff Mum, Tan Huang, who's now at Michigan. Will Talbot at Stanford shared the IRF8 mutant with us. We've also received a lot of help from Zerk in terms of antibodies and some other fish lines. And finally, none of this would be possible if it weren't for the very generous support from the NIH and RPB. So thank you all very much for the time and for paying attention. Gave me the opportunity, and I'll be happy to answer questions.
If you have any questions, just send me a chat that you have one. You don't need to write the question, but indicate that you would like to ask questions. Brian, let me start as people organize themselves. The two questions I have, or two problems to solve. One is that fish, if I remember, has many different cone types, and you were grouping them as cones. So do you see differences in different cone types as they regenerate?
So we have not specifically looked at that. Ted Allison, when he was a postdoc with Pamela Raymond a number of years ago, did look at, in wild type animals, the ability of different cone types to regenerate. And he's done a lot of amazing follow-up work with that over the past several years. But if you take a wild type animal and you do an injury, the full cone mosaic will grow back. And I don't remember him reporting, I could be wrong about this, I don't remember there being an obvious difference between any of the cones' ability or capacity to regenerate. That being said, the cone mosaic that's been beautifully described by a number of groups back in the 90s and early 2000s, that cone mosaic is not perfectly reformed. So there are some differences in the regenerated retina, but in terms of cone subtype abilities to regenerate, I'm not aware of there being any major differences.
So if you can briefly comment also, my second question is how your study and other people in your field relates to mice or mammals in general in regeneration capability, kind of like what Tom Ray published and other people in this field. Can you just briefly comment on that?
Sure. So there are some fundamental differences. It appears, so one of the first things that appears to be different is the role of notch signaling. So whereas notch signaling, I hope I don't mess this up, notch signaling is relatively high, I believe, in zebrafish and wild type animals, and then it decreases fairly rapidly following injury. And this is helping those Mueller cells to maintain their quiescence in a wild type animal. In mice, it's sort of the opposite. In notch, modulating notch in a mouse does not appear to have a major effect on the ability to regenerate. Similarly, the role of microglia appears to be somewhat different. Tom Ray's group recently found that if they ablate microglia, they actually enhance the ability to regenerate in their injury paradigm, where they overexpress ASCL1, they treat with NMDA, add a histone deacetylase inhibitor, things like that. So that being said, the types of regeneration that you can obtain from mouse versus zebrafish are somewhat different. The proliferation capacities are somewhat different. I found it personally a little challenging to try to make a direct, apples to apples comparison, just because there are a lot of differences in experimental approach. That being said, I think you do see a lot of conservation in some molecules, and you see some pretty fundamental differences in some other things, namely like notch and the role of microglia. So I think there's still a lot to learn about what are some of the species differences. And then you've got to throw in all the work from Andy Fisher on chick, which also differs a little bit in their ability to regenerate.
Thank you. Dorota.
So you partially answered my question. Well, like you said that you didn't study it too much, but I wanted to still ask about one thing. How would you address the fact, the problem of maintaining the cones after regeneration in the diseased retina? So how would you address that point?
So I think it's sort of it's a good question, and it's an interesting question. So if you regenerate, and if I'm understanding kind of the basis of your question correctly, you're regenerating from endogenous stem cells, which is what these Mueller cells would be. They're still going to carry the same mutation that was present in the photoreceptors to begin with. So these are still going to be mutated photoreceptors that will very likely die. And so are you going to have to continuously regenerate these because they're always going to carry the same mutation and ultimately die? I think that's a problem that's going to have to be worked out. There may be a combination of regeneration along with some kind of a gene editing approach to hopefully correct it. If you've already started with a retina that's largely depleted of photoreceptors, and you have to regenerate those anyway, you still may want to correct those. In other cases, maybe you have such a slow degeneration that if you regenerate them once in a human, if we're going to think very futuristic like that, maybe that's sufficient to last through the lifetime of that individual. And if it's a slow enough degeneration, maybe once is enough. Maybe we find other ways of preserving photoreceptors, whether it's through immunosuppression, targeted immunosuppression, things like that. At least with our results, suggest that reducing microglia was protective on cone degeneration. So I don't know that there's one direct answer. It may be a combination of things, but it is something that's very important to consider.
Thank you.
Wonderful. We have several questions, and good news is that Susan will make a final comments after those. All right, and bring her question. All right, John.
Thank you for a good presentation. I have two questions. My first question is related to what Dr. Palczewski asked about zebrafish versus mice. Obviously, zebrafish being also a good model, being a vertebrate. Have you come across or have insights into like where this regenerative capacity of Mueller glia got maybe lost or limited over the course of the evolutionary tree from the zebrafish going up to a mammal? And yeah, that's my first question.
So I'm sure there are probably others listening in that would have a much better answer to this than I would. Certainly, I have not done a deep enough reading to know all the different types of animals. I know that certainly, zebrafish have a really good capacity to regenerate. Things like newts and salamanders show a fairly robust ability. As you go up the evolutionary tree, chick has a good ability to regenerate. Very, very young mammals have a pretty good ability to regenerate following damage, but as they turn into adults, then that capacity is far more restricted. Given that we haven't sampled all the different species that are out there, it may be possible that there are some mammals that have slightly better abilities than laboratory mice, but I honestly don't know enough about the literature to really speak intelligently and give you a really good answer for that. Sorry about that.
My second question is going back to the cytokines and how different cytokines get elevated very quickly and then sort of diminish, and then other cytokines get elevated later on and then diminish, analogous to the processes of wound healing. Have you profiled or have you tracked the timing at which these things get released in zebrafish to facilitate this regenerative capacity from Mueller glia?
So we have not in my lab, but other labs certainly have. It's one of the things that's on the to-do list, probably the next couple of months. But yeah, so both Dan Goldman, Peter Hitchcock, David Hyde, a lot of the other groups have certainly done good profiling of what happens. And certainly, between David and Seth's really elegant science paper a couple of years ago, where they did single cell analysis at multiple time points after injury, there should be a fairly extensive catalog now of all the different inflammatory cytokines, both pro-inflammatory and anti-inflammatory, that are released and the timing of those. So it's fairly well documented that you get an early peak of IL-8, within the first 8 to 16 hours after injury, and within about 2 days or so, you have a large increase, or 1 day, I think, and that continues on. You have a big spike in TNF alpha, TNF beta, and then the anti-inflammatory cytokines come on closer to like 4 days after injury. So there's a fairly extensive list. Now, how that differs in acute injury versus the kinds of inflammatory cytokines that are expressed in the mutants, I think is still something that we need to dig into a little bit deeper.
Thanks, Emma.
Yes, so my question is on the last part of your talk, and I was wondering, in the transcriptomic studies of the Mueller glia, was it your impression that they're a fairly homogeneous population, or were there certain distinguishable characteristics between Miller glia that were more or less amenable to reprogramming and regeneration?
So the work that we did where we found Notch 3, it was relatively preliminary, and we didn't have a really large sample size. We're gearing up to do a much more robust profiling of those cells, and I'm hopeful that we'll be able to have an answer to that question, hopefully another year and a half. What we can say at a very 30,000 foot level is that if we look at those Mueller glia and then basically overlay their profile with that from the previous study where they looked at wild type Mueller glia, both uninjured and over the first several hours after injury, the Mueller glia in a Sep290 fish was distinct. Now, exactly what was being expressed in those cells and did they have certain characteristics, we really couldn't say at this point in time, but they do seem to be distinct.
Emma.
Hi, Brian. Nice
Hey,
to
Emma.
Dr. Enrico last week, I know Dr. Beata has been also working on inflammation in relation to degenerative diseases, but the work that they have been mainly doing is local, right there in the eyeball. Here with your zebrafish model, you're actually doing it systemically. Do you think that peripheral immune cells might be playing a role in the degeneration that you are observing in the mutant? Have you investigated for that?
So we have not looked directly at that. We rely a fair bit on the work of Jeff Mumm. And he found and reported a number of years ago that there does not appear to be any real infiltration in the zebrafish retina, at least, of circulating macrophages into the retina. So what we think is occurring, even following our acute injury paradigm, and I should say that a needle poke or something else that's going to damage the blood retinal barrier, that's a different story. Our light damage experiments do not appear to have any effect on that. Everything remains self-contained. There doesn't appear to be a lot of migration of peripheral macrophages into the retina. So what's happening is most likely a local event. It's possible, and we do think that there might be some migration of microglia through the optic nerve into the retina, but that's still going to be somewhat limited. So I guess to answer your question, we don't think that there's much effect from peripheral or circulating immune cells to what we're seeing. But it's an interesting question, something to certainly consider.
Don, all yours.
I have a question. Oh, sorry. Is someone going?
Don's active. So
Yeah.
right.
Okay.
Sorry, Chris, I couldn't hear when you said Don. So hi, Brian.
Hey, Don.
Great talk. Thanks. So I think you and Jeff and a number of people really elegantly showed the importance of, inflammation, both, in cell death and regeneration, but inflammation obviously is a very complex process, and your IRF7 studies were cool. Is there any combined effort in the zebrafish community, given the power of zebrafish for genetics, to really dissect in a more coherent, and the gene expression studies you cited obviously have prioritized certain candidates, but is there a coherent effort to really dissect more what's going on in inflammation, and would some kind of interaction be better to do it in a more systematic way?
So is there a systematic approach? Not that I'm aware of. I think that all of us, obviously a couple of weeks ago, I came back from the ICER meeting, and what, Jeff Mumm said, what Jeff Gross said, I shall repeat, which is we've all basically got dragged kicking and screaming into the immunology field because it was something that we were terrified of as graduate students. And so the ability to have, I think, one of the advantages of mice obviously is the ability to rely on 30, 40 years of really well-developed markers and antibodies and things like that. And I think there's, a fair bit of conservation between zebrafish and mice in terms of like just the immune response, but there are a lot of genome duplication, for a lot of these immune genes, and just the ability to have, good markers and using the genetics approaches to make mutants and the necessary transgenes and everything else. I'm sure that there would be an interest in it. We are still a relatively small group of people, but maybe harnessing combined efforts would be useful. Picking appropriate targets. I'll put a shout out to Diana Mitchell. I mean, she's putting a lot of work on, purifying microglia and macrophages and trying to get really comprehensive catalogs of everything that they're expressing during injury. So that might be a place to start, at least to then develop the next set of tools to try to go after some of the things I think you're alluding to.
So 2 more questions from the room, and then we'll go to Susan. So let's start with Emily.
Hi, Dr. Perkins. Very nice talk. I want to preface this question with I'm somebody who's never personally worked with zebrafish, but I would like to ask if you have, or are there ways to check the function of these regenerated cones in your mutant, not my zebrafish.
I'm sorry, I missed the middle part of your talk. Do we have a better way or a different way of analyzing cones after regeneration? Is that correct?
Checking the function of these cones and how they are compared to either the normal or not damaged.
Yeah, so that's a great question. How can we test function? So I see Sue on the line here. She might know a little bit about this as well. But you think about an ERG, and that's still a full field response. And since the light damage experiments only ablate maybe somewhere between 30 to 50% of the cones in the dorsal part of the retina, you're still leaving the majority of the photoreceptors in the retina largely undamaged. And that would maybe show up on an ERG, but that's a full field response. And after the regeneration occurs, you'd have to really be trying to find somewhat subtle differences to really pick up anything. So that leaves you really with probably single cell recordings, which would be pretty challenging. You'd have to find a way to distinguish. I mean, again, we track all of these predominantly using EDU, but doing it in a living retina, you'd have to kind of guess a little bit as to where those regenerated cones were, unless there was a way of somehow marking them genetically, which there could be. There could be ways of doing that. There is some evidence that, it's a pretty compelling and strong evidence that these regenerated photoreceptors do make functional contacts, they make functional synapses, all the necessary proteins for synaptic connections are present. There appears to be recovery of visually driven behavior in these animals after, from after-light injury and then after regeneration. But I think, what you're asking is an important question, which is, can we get a little bit more rigorous and ask, are these regenerated cones and rods maybe more electrophysiologically normal, or are they different in some way? I don't have any reason to suspect that they would be different, but I'm not aware of any evidence that they have been directly tested like that.
Hi, Dr. Perkins. It's Dominic Lewandowski. Great talk. I have a question regarding IRF8 mutation. So in this mutation, maybe I missed that, but did you check the microglia numbers before and after the damage, or only undamaged retinas? Since we know that there could be recruitment of microglia, macrophages to the retina and proliferation of microglia, so they could still play a role in the regeneration if they get recruited from the bloodstream.
Yes, that's a very important point. I didn't show that. We have we do have that data, or gathering that data. We have looked at slides. It's not been quantified rigorously yet, but we do have experiments that we are still basically analyzing currently, looking at co-localization of microglia markers with EDU and with PCNA at different time points immediately after injury. Just qualitatively, we see there is a little bit of overlap, but there's not a lot in the IRF8 mutant. So there doesn't appear to be a significant amount of proliferation of the residual microglia. There is a little bit of an increase. Again, it's not quantified, so I can't give you exact numbers. It does not appear that there is a huge increase in infiltration of microglia. Again, certainly not from the outside. There might be some coming in from the optic nerve. We see probably a little bit higher concentration of microglia down near the optic nerve. But it doesn't appear that we see like a tenfold increase after light damage, either due to proliferation or from infiltration of circulating macrophages or microglia.
Very well. Thank you, Brian, so much. And now the final comments, questions go to Susan.
Yeah, so I just wanted to follow up on sort of what you've already said a little bit. So I thought it was really interesting that deleting the microglia rescued the CEP290 cones. So many of us have always thought that, you know, it's the intrinsic defect in the cell that's causing the death, but this sort of suggests it's both the intrinsic defect and the interplay between the intrinsic defect and the microglia. And so zebrafish are really, as you've in your recent review have written really great as a model for studying retinal degeneration. And there are a lot of different retinal degeneration mutants. So I just wonder if you are planning to sort of follow up on that to see to what extent suppressing the immune system could rescue degeneration in all the other disease models.
Yeah, it would be a really great project. If I can get some more money and some more people, I think that would be really fascinating to try. As you're well aware, something like, you know, I think there's a big difference between looking at a protein like CEP290 that, you know, by all accounts doesn't have an enzymatic function. It's probably a structural protein that's helping coordinate the assembly of this transition zone within the cilia, as opposed to something like, you know, PDE6, which, you know, obviously as, you know, you make a mutation there, you have catastrophic consequences on, you know, calcium levels and everything else in the photoreceptor. And so the ability to rescue cone survival, it's probable that the cones are just unstable and a little bit unhealthy from the intrinsic defect in CEP290. And maybe that's causing some kind of, you know, neurodegeneration, you know, due to elevated inflammation. And so by lowering that, you may help preserve them a little bit longer, where the extrinsic challenges are maybe overwhelming some of the intrinsic problems. I think the opposite may be occurring in like one of your mutants in all likelihood. I don't know that IRF8 deletion would rescue those, to be honest with you. I mean, maybe it would. I think it'd be really cool to try, but we were surprised as well. And I think it could be just the difference in the types of mutants that you look at, as how much of a role does the intrinsic mechanisms play as opposed to just kind of a general inflammatory, just stressed out nature is driving the cell death.
Brian, thank you so much. That was wonderful show this morning. And
thank you, Chris.
Next time.